We have now moved on to the most advanced single-celled organism, the protist. Before we can talk about the protist, we have to spend a little bit more time talking about classification and extension of taxonomy. The taxonomic hierarchy was revolutionized by the advent of phylogenetics. Phylo meaning family, genetics referring to the actual genetic sequence of the DNA of these organisms. By applying phylogenetics to taxonomy, we were able to build genealogical trees based upon shared evolutionary features. In doing so, we were able to build connections between groups that had otherwise only been inferred. The result was a cladogram, a branching diagram showing relationships between species. Cladograms are sometimes referred to as phylogenetic trees. Just like real trees, the longer the branches, the older they are, the lower they will appear on our evolutionary history track. Each fork in the road, each branch, if you will, depicts a distinct group, a unique adaptation or gene or trait or mutation. So we can look here at this particular diagram and see that every V shape is where one group goes one way and another group goes a different way. So as we track this diagram, we can see that all organisms in this tree have a spinal column. That's the oldest, most basic general trait within this group. But what separates brown bears out from all the other bears is the shape of their cranium. What separates the sun bear out from the spectacled bear and the raccoons and dogs is its nose. What separates out a panda from dogs is the type of ear shape, round versus floppy triangles. The giant panda versus the red panda is divided based upon fur type and color. Meanwhile, the raccoon versus dog depiction is divided based upon the tail structure. Be comfortable recognizing the concept of a phylogenetic tree and navigating these left versus right differences in gene acquisition. You might recall a cladogram is a phylogenetic tree, but how do you read this tree? Well, by looking at the branching patterns, it became clear that there are three important relationships that can be depicted by these cladogram structures. Be able to recognize the difference between monophyly, paraphyly, and polyphyly. Monophyly, the prefix mono describes one. A monophyletic family is one complete family, the shared ancestor and every descendant then on. Paraphyly, para, meaning kind of, sort of, describes an almost complete family. And poly, the prefix meaning many, refers to more than one family being grouped together. Let's take a look at these in turn. Monophyly would highlight in this grouping of purple the ancestor Sauropoda and every branch that came after that. Another example of a monophyletic grouping would be this gray structure, which includes the ancestor Tetrapoda and every ancestor after that. So these groupings can go back as far as you need them to or be relatively modern. As long as the shared ancestor and all their descendants from that point forward in time are included, we maintain a monophyletic family. This is in contrast to a paraphyletic family, which would be almost complete. In this case, we see the ancestor Reptilia is included and almost all descendants. But the purple grouping does not extend around aves, the birds, and so we don't have a full or complete picture of this ancestral lineage. Polyphyly is the only grouping that does not include the branching V point. Instead, the purple grouping extends outside the tree, grouping two groups, such as grouping both Mammalia and aves, the mammals and birds together, due to something shared, like living on land or being heterotrophs. But the reason why the purple grouping is outside the tree is because that feature evolved separately. It's not from having a same ancestor that gave rise to this. The protists are the simplest eukaryotes. Before we can discuss the simple eukaryotes, we have to discuss eukaryotes as a whole. Eukaryotes are a specific cell type that could be single or multi-celled. All eukaryotic cells contain a nucleus, which houses the genetic details of the organism. The genetic structure is many, not just one, chromosomes that fold and coil into X shapes. And surrounding the nucleus are a number of organelles that are capable of doing work, subcompartments with functionality. How did eukaryotes come to be? The theory is known as endosymbiosis. Remember that scientific theories are really observations that have been proven based upon past studies. Endosymbiosis literally breaks down into living within. Endosymbiosis describes a process by which two side-by-side prokaryotes were living in harmony until one engulfed the other. And for some reason, instead of digesting it down and eating it, one prokaryote was able to survive within the other. And furthermore, when the outer prokaryote divided, it was able to pass on the internal prokaryote to its offspring and thereby create a perpetual occurrence of this relationship. How do we know endosymbiosis is a thing? Well, we know that it came about from mitochondria and chloroplasts because those two cellular components have their own DNA. And because we have phylogenetics now and the ability to sequence genomes, we have been able to compare and connect that DNA to free living organisms that exist in the world around us with some 97-98% accuracy. Additionally, the mitochondria and the chloroplasts are double membrane, speaking to the fact that they had their own membrane prior to being absorbed and taking a second layer. Unlike all the other compartments and organelles in the cell, the chloroplasts and the mitochondria are autonomous, meaning that they can maintain homeostasis, be self-regulating themselves. If they want to divide more, they do it. They don't have to check back in with the nucleus and wait for gene expression for that to occur. All cells contain ribosomes, and it's interesting to note that the ribosomes that are contained in the eukaryotic cytoplasm are larger than the ribosomes that are contained inside the mitochondria and the chloroplasts. These ribosomes are the same size as those exhibited in bacteria. Lastly, how they reproduce. Chloroplasts and mitochondria exhibit a mechanism very similar to binary fission, without all the checks and balances and regulatory steps that we would otherwise see in mitotic division. As we move forward, it's important to recognize that the Protist group is very diverse. Within the Protists, the organisms come in different shapes, sizes, colors. Some are photosynthesizers, some are predators, some are obligate parasites. They live in a wide variety of habitats, exhibit different cell structures. It seemed that the only thing that they really had in common with each other were that they were single-celled and complex. So when taxonomy first began, all the other living organisms were divided out into their groups: plant, fungi, animal, mammal, insect, arachnid. The protists were shuffled off to the side as just one uber group known as the simple single-celled eukaryote. However, now that we had phylogenetics, once phylogenetics became an option, once we applied phylogenetics, we were able to discern the fact that there were smaller groups within the protists. We refer to these as supergroups because we had to insert this grouping inside our taxonomic hierarchy between domains and kingdoms. We're going to talk about each of these supergroups in turn and highlight what makes them unique. We begin with the archaeplastids. Their key feature is that they are all photosynthetic. They also exhibit something known as a zoospore, which you can define as a flagellated spore. It is the precursor to sperm. Their overall significance is that they gave rise to all the land plants. Next group are the chromoalveolates. They are further divided into subgroups alveolates as well as stromatophiles. The big thing to recognize here is that the alveolates have a membrane-enclosed sac beneath the cell membrane, so they have an extra layer. The stromatophiles have hairy flagella. Overall, the chromoalveolates serve as the base of our food chain. Our third group are the excavates. The excavates' key feature varies depending upon their subgroups, although all excavates have flagella. The diplomatids have mitochondria, but they are non-functioning. The parabasalids have semi-functioning mitochondria, and the euglenozoans are able to do both photosynthesis and cell respiration. We refer to this as mixotrophy. Another key feature of the excavates is that they are asymmetrical organisms that tend to look like commas. Next up, we have the amoebozoas. Their key feature is that they move by pseudopods. The term pseudopod describes a false foot. Basically, they have these cytoplasmic extensions that reach out and then contract, pulling the rest of the organism with them. And so they tend to exhibit a reach, grab, and pull, almost crawling motion across the ground. Their significance or role in ecology is that they are prolific decomposers. The Opistheconta are our next group. Their key feature is that they have mitochondria, modern mitochondria, and a single posterior flagella. Their significance is that the Opistheconts gave rise to the animals and fungi. Our last group are the Rhizaria. Their key feature is that they have these needle-like dried pseudopods, and their subgroups are broken down based upon what those pseudopods are built out of. If they are calcium carbonate, like our skeletons, they're from the foramens group. If they are silica, like glass, they belong to radiolarians. These groups are used in carbon dating. Among all the different protists that exist, malaria is perhaps the most important and significant. In terms of public health, malaria is considered a top 3 global health concern. It is a tropical disease, meaning it occurs between the tropics of Capricorn and Cancer. In this area, half of the world's population reside. And as a result, roughly 1.2 million people die every year from malaria and malaria-related complications. The malaria parasite is only transmitted by Anopheles mosquito. The Anopheline group is one of 3 mosquito genera. There are Culex and Aedes that transmit viruses and other bacteria, but Anopheles is the only one that can transmit the malaria protozoan. And within the group of 400 approximate species, only a small proportion, 60 of them, are capable of passing this pathogen on. And so it's really surprising to see how prolific the malaria infection rate is annually. Let's talk about the parasite. There are 5 species that infect humans. I have them ranked here from the most severe to the least severe. Plasmodium falciparum is responsible for death 99.9% of the time. Thankfully, Plasmodium vivax is the most common that we see worldwide. Plasmodium malariae and ovale are really rare, and we usually only see them when someone is co-infected with something else. And Plasmodium knowlesi is relatively new. It emerged in about 2010, and so far there've only been about 20 human cases. Previously, it was in macaque monkeys. Why is it that one species, Plasmodium falciparum, is able to account for 99.9% of all malaria deaths? What makes that particular species so much more severe than its 4 other competitors? Well, for one, it's the only species that can invade the spinal cord and brain, and thereby, if it reproduces there, cause massive tissue damage. Also, it's the only species that invades both old and young red blood cells. All the other species prefer to infect old red blood cells. And so when those blood cells die during the replication process, the body was prepared to replenish them anyway in short order. And so the human host is not working at a deficit for long. However, if a young red blood cell died, the human body was not prepared to replenish that for, say, about another 3 months. And so the person infected with falciparum may be operating with less red blood cells, which means less cell respiration and increased fermentation for a whole slew of additional side effects, typically relating to pain, fatigue, and arthritis. Lastly, it has the fastest reproductive cycle. It reproduces every 24 hours. And so, taken together, these three things result in this one species being the one that's fatal if untreated. What does malaria look like? Well, on the whole, it is basically a super flu-like disease characterized by relapsing cyclical fevers and chills. If you were infected with malaria for a long time, complications arise when a person's infected with malaria for an extended period of time. Chronic infection can lead to spleen enlargement, liver and kidney failure as a result of trying to filter out ruptured red blood cell debris, and brain damage in the event that you were infected with falciparum and cell reproduction occurred there, causing tissue damage. The mortality is estimated at 1.5 to 2.7 million deaths every year. It's interesting to note that malaria is not something that you can get person to person. The mosquito is key. And while we can talk about malaria in terms of what's happening in Brazil or India or Africa, the truth is the US has a history of malaria as well. In fact, we used to have 6 million cases alone. The Center for Disease Control was actually created and originally titled the Office of Malaria Control. Its vision was to combat malaria first and foremost. And once it was able to do that through construction, drainage, and civil engineering, it was rechristened the Center for Disease Control to then tackle other infectious agents. Now, while the parasite is no longer within our borders, the mosquitoes continue to live here. These are vector species. And so occasionally we see local outbreaks because a traveler has brought back the pathogen, and if a mosquito feeds on that infected person, it's only a matter of a few weeks before that mosquito could go on to pass it on someplace else. This raises concerns about climate change and global warming because as the temperatures fluctuate, we might create a more favorable habitat to sustain the parasite's life cycle, and that might not limit us to local outbreaks anymore. Malaria is one of the older Protist species, and so we see a long history of human disease. There's a minimum of 10,000 years of recorded history that have identified malaria-like illnesses in humans. In fact, King Tutankhamun, the young teenage pharaoh, died of malaria, and you can actually recognize the parasites wrapped in his mummified remains. And so, given that long history, some human populations have developed resistance over time. Sickle cell anemia is a disease where a single nucleotide mutation resulted in a single amino acid substitution, and this altered the red blood cell shape. Normal red blood cells should be happy little donuts that can carry and ferry around lots of oxygen. But the sickle cell anemia red blood cell is flat and sickle-shaped. This reduces the amount of hemoglobin and therefore oxygen it can carry. Now, if a person got 2 bad copies of the sickle cell gene, they would die within the first 2 years of their life from multiple organ system failure because there wouldn't be enough oxygen to go around and support the internal workings of the body. Here in the United States, we see almost no occurrence of sickle cell anemia because if you had 2 copies, you would die. If you had 1 copy, you would live, but typically those individuals only live to about 35 to 40 years old due to additional heart strain from having to work to pump and filter out these flattened red blood cells. Additionally, those individuals are considered less fit in life because they cannot use cell respiration entirely and therefore have to rely upon fermentation, which is an added toxicity to the cell cycle and burden. Yet, when we look at other places in the world, particularly Africa, we see that the sickle cell trait, being a heterozygote carrier of the sickle cell trait, is the most common, the most preferred outcome. And the reason for this is, in Africa, if you had the sickle cell trait, 2 copies of it, you would die like anyone else. But if you had 2 copies of the normal red blood cell trait, you would likely get malaria and die by the time you were 5 because falciparum is so prolific there. However, If you happen to get one good copy and one bad copy, and therefore were heterozygote, having two different forms, half your blood cells would be donuts, half of them would be small sickles, and this allows your body to get just enough oxygen to keep you alive, but not have any extra oxygen for the parasite. And so those who are heterozygote for sickle cell anemia, even though this is not ideal in the long run, it is considered a heterozygote advantage because they are now resistant to malaria infection. Again, they won't live to the normal human life expectancy of 86 years old, but living to be about 40 is preferable when the other options are 5 or 2. The last two prokaryotes. Let's talk about two other protozoan parasites, the trypanosomes. These belong to the excavate group. It's interesting to note that these trypanosomes are genetically identical, but because they evolved in different environments, they have adapted to two completely different vectors and result in two different diseases. In Africa, the trypanosomes cause something known as African sleeping sickness, which makes a person so very tired that they're likely to sleep and sleep and sleep. This is not a common disease. Only roughly 10,000 people are infected annually, and it's not likely to be fatal. These trypanosomes are passed on by the tsetse fly. What makes these interesting is that the tsetse flies are very small, so they can usually penetrate a mosquito net, but more importantly, they are out during the day, during a time when you are walking to work or playing at school or working in your fields or gardens, and therefore not under a mosquito net. In the Americas, however, the trypanosomes have caused a disease called Chagas disease. This is interesting because it infects roughly 10 million people every year, and it has been recorded all the way down in Argentina, all the way up through Canada. These trypanosomes have adapted to a different vector, the kissing bug. These bugs feed on you at night, and they tend to bite on your lips, the soft tissue there, and thereby get the name kissing bugs. They nibble on your lips, causing small blood, and through this, the trypanosomes can enter. This also is not a disease likely to kill a person, but it can cause flu-like symptoms, fatigue, and irritation.